All right. Good morning, everybody. Welcome to our weekly medical grand rounds on this blistery morning that winter decided was going to come back yesterday, at least for a day or two. So before we begin, I would like to respectfully acknowledge that we are located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to enrich our vibrant community. So I'm delighted today we have our regularly scheduled research rounds, and I will ask Dr. Gopi Satendra, who is one of our directors for the graduate program, to introduce our speaker.
Thank you, Norman. So today it is my great pleasure to introduce Dr. Lara Mahal to give her talk today on glycosylation. Just a little bit of background. Dr. Mahal, she had obtained her PhD in chemistry at UC Berkeley in 2000, and she was the first student of Professor Carolyn Bertozzi, who was the Nobel laureate in 2022. She then trained under another Nobel laureate, Dr. James Rothman, doing her postdoctoral fellowship in Sloan Kettering Institute. From there, she had her independent academic position in chemistry at the University of Texas and then at the New York University, where she was a faculty member till 2019. So she was then recruited here to the University of Alberta in the Department of Chemistry with the Canada Excellence Research Chair and Glycomics. And as most of you know, this is one of our most prestigious chairs, definitely for recruitment. Dr. Mahal has published, well over 100 papers in the field. And I just want to make a point that we're really honored to have her give the talk here because she collaborates with a lot of people here in the Department of Medicine. A lot of the work she'll present, although she's in the Department of Chemistry, it's very interdisciplinary to our clinical to our clinical academics here in the department. For example, she collaborates with people from the Division of Infectious Diseases, Cardiology, and Rheumatology, showing how glycosylation is important in influenza. I believe she'll be talking a little bit about that, but also a lot of other clinical diseases that we study. And really the importance of these grand rounds is to have interdisciplinary collaboration. And so we think that the work from Dr. Mahal will really initiate that, and we hope it'll initiate much more collaborations because that's something she's also involved with. She collaborates heavily, and a lot of the work she does is of great impact to what we do here in the Department of Medicine. So thank you, Dr. Mahal, for giving this talk, and welcome.
Thank you so much for the invitation. I'm really, really honored to be able to come and talk to you about our work. And let me say, for those of you who actually made it to the physical audience, thank you so much for actually braving the renewed winter onslaught. And for those of you on Zoom, I completely understand. So I'm going to talk to you about our work in glycomics and of course, I might, because it's decided that it's not going to work anymore. Okay. And apparently it's not working on any level. Yay, we spent half an hour trying to fix all of these things, and then it decided to go to do this right as I was. It's lovely. Well, so I'm going to do my disclosures while we're waiting. So I only really have one disclosure, which is that I consult off and on for Vector Laboratories, which is a lectin company. And you'll kind of see why they asked me to consult in a few minutes as I talk about it more. The keyboard works. Okay, great. Yeah. Oh, no, now this is working again. Okay, I'm going to skip the land acknowledgement. Okay, so as doctors, I think one of the things that people have heard a lot about is precision medicine, right? And if you think about precision medicine, you think about the fact that standard therapies don't work for everybody, right? There's not really a one size fits all. There are a lot of different reasons for that. It can be biological sex. It can be because you have mutations that don't make one drug you as well as another. It can be for a lot of different reasons that have to do with your metabolic health, et cetera. And so there's been a real push to get to precision therapy, to try to get more personalized medicine, to try to get to a point where we can use things like omics and health data. What are you eating? What's your metabolic health? What is your state? to be able to get to tailored therapy so that we can get really good therapeutics, so that we know that when we give somebody something, they're going to get better, right? So how do we get here? Well, part of this is really data-driven, right? We need data that gives us information that allows us to figure out what's going to work for who, right? And that data right now, in terms of the omics, I mean, other than the standard health data that people gather, we're often using omics, right? So we use genomics, we use transcriptomics, we use epigenetics, we use metabolomics. Metabolomics are already being used in childhood diseases to figure out if people have particular childhood defects that need to be fixed, right? So this is a huge part of the panel that people already are using to try to get to this personalized medicine. But what about the glycome, right? And so the glycome may be a new word to all. So I am going to spend a few minutes doing a little bit of definition because most of you probably don't remember some of the things about sugars, which is fine. So the glycome is the collection of complex sugars or glycans in your body or in a tissue. And so I'm going to have to show you a little bit of organic chemistry. So I apologize. As an OChem teacher who taught pre-meds, I know how scary this is. And so this is the first and last slide that will have these 6-membered rings on it, not to worry. And the reason I'm showing it to you is because I want to point out that glycans are made from multiple different monosaccharides. One of the ones you're probably the most familiar with is glucose, right up here in the corner. I want to point out that throughout the talk, I'm going to be using the really cool Lucky Charms version of these, which is the symbolic nomenclature for glycomics, which carbohydrate chemists and biologists have actually agreed upon, so that we can stop showing you those scary 6-membered rings, right? And actually get to something where you can recognize the patterns. So these are the 10 common monosaccharides that one sees in mammalian sugars. Things like sialic acid are really critical in cancer. There are new drugs that are actually in clinical trials to try to shave off sialic acids to make immunotherapeutics more effective. This is also the sugar that influenza binds to, and the target of Tamiflu. So, there's galactose. So these are all a sugar code. And given the time, you guys are not going to remember it, but that's okay because I'll tell you as we go along. One of the things I want to point out is that the way these sugars are connected matters. So these aren't, when we talk about these, we're going to talk about complex structures that are connections of all of these, right? And the way that they're connected matters. And this is showing you the chemical representation, and this is showing you the SNFG representation. So for example, beta-1,6 branching of an N-glycan, and I'm just showing you that trisaccharide part of it, is really important in multiple sclerosis. We'll get into that in a few minutes and in some other areas. And it's bound by a lectin called PHAL, which does not recognize beta-1,4 branching. So when you look at a triantennary N-glycan, if you ever do, which none of you probably ever do, which is fine, those are actually potentially two different structures, depending on whether it's branched on the 6 position or the 4 position. And so I point this out because sometimes these things get lumped together, but those differences in structure are actually meaningful, and they're biologically meaningful, and they have importance in terms of how these things work. Okay, so, and I also want to point out that these complicated sugars are actually on a variety of proteins. So many of the antigens that you look at, whether you're looking at transferrin, right, in the assays that look at glycosylation on transferrin as a marker for congenital disorders of glycosylation, whether you're looking at the prostate-specific antigen, right, which is very commonly looked at for prostate cancer, many of those proteins are glycosylated. In fact, about 80 to 90% of all cell surface or secreted proteins are glycosylated. So these sugars can't ignore them because the glycosylation of the proteins may have really important biological information that may impact the way that those proteins work. And in fact, we know that they impact the way that proteins such as antibodies work, which given the number of antibody therapies that are coming out is kind of important, right? So we're starting to see people understand that sugars are not something we can really ignore. Right? These glycans are not something we can really ignore when we're starting to think about how things are working in medicine. So I want to point out, like, there are glycolipids. These are actually lipids that have sugars. Most of your membranes are coated with glycolipids. GM1, for example, is an example of a glycolipid. This is actually what cholera toxin binds to, right? So when you have cholera and you have like all of those illnesses, it's actually what's been bound in the gut. On the plasma membrane, you have N-linked glycans. This is an example of an N-linked glycan. You also can have O-linked glycans on mucins. An example of a mucin you may be familiar with is CA125, right? So these are O-linked glycans versus N-linked glycans. There are also other structures. Also glycosyl aminoglycans and proteoglycans, so things like chondroitins. Probably the most famous glycosyl aminoglycan that clinicians are familiar with is heparin, right? So you're actually already using in medical practice sugars. You just don't realize it because you never think about the fact that they're sugars, right? So, okay. So I want to point out that glycans are already informing clinical decision-making. So it's not really a big stretch to think that you might want to think about these in, as a marker for clinical decisions. Here's a basic example, blood type, right? Your blood type is encoded by sugars. When you give someone a blood transfusion, you have to consider the sugars on their cells, right? In order to be able to give them the proper blood type. When you do a transplant, you have to consider the sugars on their cells and whether or not people have antibodies to those sugars in order to get appropriate transplantation, right? So you're already using, at the most basic level, sugars in your decision-making, but you just don't realize it. Another set of things where we know sugars are involved in clinical decision-making are cancer markers. So CA19-9 is commonly used in panels to look at progression of pancreatic cancer, right? CA19-9's name, the scientific name for it, is sial-Lewis A. It's actually a very famous carbohydrate antigen. A related antigen, and you can see that these are actually very, very closely related. This is a beta-1,3 linkage. This is a beta-1,4 linkage. I'm just pointing this out because literally the linkages are the only things that are different between these two. This is CD15, which is often used to look at blood cancers. That's sial-Lewis X, right? So sial-Lewis A and X, or CA19-9 and CD15, as you might be more familiar with them, are already in clinical use as biomarkers. Another famous biomarker is CA125, which is actually, we are not necessarily looking at the glycans themselves, although that does influence how the antibodies are binding to them, because the part of this protein, so this protein is a mucin, and all mucins are heavily, heavily O-glycosylated. That's what the definition of a mucin is, right? So every mucin in your body has a very heavy O-glycosylation. And in fact, it is that O-glycosylated portion that is actually part of what the epitope is for CA125. So depending on the antibodies that you have, you might be seeing different glycoforms of that particular protein. And just to give you a definition, a glycoform is a specific carbohydrate, so a specific protein with a specific set of sugars on it, right? So you can have different glycoforms of the same protein, right? Okay, so this is just a little bit to show you that this is one of my standard slides. Glycans are actually involved in the physiology of pretty much every disease. And most people are just not aware of it because they never really think about it. From the simple, so when you get the flu, you are the victim of a carbohydrate interaction. So we have alpha-2,6 sialic acid in our upper respiratory tract. That is what influenza binds to. The reason we do not currently get bird flu is that bird flus bind to alpha-2,3 sialic acid, which we have in our lower respiratory tract, but we don't have as much of it in our upper respiratory tract. Pigs have both. It's one of the reasons that they talk about when ducks and pigs are close together, you start to get emerging influenzas that can actually go into humans because pigs have both. And so they're a species that allows the adaptation of that virus from the bird into the human. Right? And this is one of the great fears with the new bird flu, right? Is that it's going to adapt to actually bind to 6, and then it's going to be easily transmissible to people, right? So that's just one example. Tamiflu, the reason that Tamiflu can only work when you give it early is that it inhibits what's called a neuraminidase, which is the enzyme that takes off that sialic acid and allows for the virus to be released. That's one of the thoughts as to what the neuraminidase is doing. That only works when you give it early because it's part of the propagation step of the virus, right? That's why you can't give it later. It doesn't help anything once you're already infected. Once that infection is established, it does nothing. And that's all because of the carbohydrate or the glycobiology of it. So you also see carbohydrates really involved in bacterial cell interactions. So when people have urinary tract infections with E. coli, They actually are, E. coli binds a carbohydrate called hymannose through a lectin called FIMH, right? So that's one example. And in fact, people are developing, and I think there's actually clinical studies right now going on inhibitors to FIMH to be able to prevent urinary tract infections. But we also see these so it's not just all about infections, right? We also see these interactions as being very important in the recognition by our immune system of cells, right? A large part of our immune system actually revolves around sugars. So those of you who think about the complement cascade, right? The lectin-mediated complement cascade. Lectins are carbohydrate binding proteins. They all revolve around the fact that our innate immune system is incredibly sugar-driven, right? Some sugars, like sialic acids, actually inhibit our immune system, and some sugars, like hymannose, actually activate our immune system. And this allows our body to recognize when there's a problem, when there's not a problem, right? Friend versus foe. But also we see this in natural cell-cell interactions. So when you have, when you are making different tissues have different carbohydrate codes. When you have interactions that are happening in cancer, you actually see those change as the nature of the cell itself changes, right? So these are really markers of who a cell is and how it is doing, right? And I think of it as the integrated information, because sugars actually require metabolic information. They require a complex pathway. So they're taking a lot of genetic and transcriptomic information. It's really all of that information together out at the cell surface where we can see it, or secreted from the cells where we can see it, right? So what does my lab actually do? So my lab uses systems approaches to decoding the glycome, and we use a technology called lectin microarrays. And as I said, lectins, such as the innate immune lectins in your body, but we actually are using more plant lectins, are carbohydrate binding proteins. And they're actually commonly used in histochemistry. So what is a lectin microarray? It's a very, very simple idea. It is an array of carbohydrate binding proteins where literally each of these different spots is well, we have triplicates, so they're not all different, but every 3 sets of spots is a different carbohydrate binding protein, right? And we literally array them on a solid support. And in our lab, every slide has about the ability to look at 90 samples. So we can actually do large numbers of samples very easily. How does this work? Well, we can look at anything. And as you'll see in my presentation, we really have. I'm going to focus on the clinical work that we've done. So I'm not going to tell you about like animal tissue and all the other things we've done. But you can look at FFPE tissue. You can look at sera. You can look at nasal fluids. You can look at synovial fluids, cervical vaginal lavage fluid. These are all things, by the way, we've actually studied. Urine, fecal matter, all of these things can be looked at using the array. We actually need quite small amounts of material. So for sera, usually I ask people for 50 microliters, and that's actually way more than we need. And most of that is because that allows us to do follow-up studies. Quite frankly, we need about 10 or less. And yes, microliters, not milliliters. So we use very, very small amounts. We extract the glycoproteins. The proteins have lysines and terminal amines. Using those, we actually label the proteins with NHS esters that allow for amide bond coupling. So basically, it allows us to make these things glow, right? So we make them fluorescent, we make them glow. And we do this in 2 colors. So our sample is in 1 color, and then we have a reference. So all clinicians know you need the reference levels, right? You need to refer to something. So we need a reference because that allows us to actually get a much better idea of the relative level of change in any particular sample set. Because these are very complex and because glycans, we can't really make a standard reference using artificial means, we use what's called a biological reference, which means that either for serum samples, it's the NIST standard. For other things, it's usually a little bit of each sample in the mix, right? So we can actually directly index things against one another. We add these in equal amounts to our array. We allow it to bind, then we rinse and we scan, and this is what an actual array looks like. And then from that, we get heat maps and information that look like this, and we can annotate those to get specific structures. So this is real data from our mole cell proteomics paper on pancreatic cancer, and this is actually the KC mouse model of development of pancreatic cancer as it develops pancreatic cancer. And you can see that there are very, very clear changes. Like you don't have to be scientists. Oh, that's going from red to blue, right? So clearly, as these tissues are transforming, the sugars are changing. So how do we know what's on there? And this is just saying like, yes, we actually do know what the lectins bind. I actually wrote the paper that has now become the most cited paper in this area, because what we did is we took our lectins and we bound it to a carbohydrate microarray from the Consortium for Functional Glycomics, where they had 611 sugars on a slide. And then we bind the lectins to them, and we can see from the information there what's being bound and what isn't, and use that to figure out what sugars are actually present, right? And what sugars are bound by specific lectins. And so we use machine learning with Daniel Bohar to actually figure out the specificities. And those of you who are interested in using lectins for histochemistry might want to check out this paper, because we have a detailed annotation of all of the commercially available carbohydrate binding proteins, right? Where we actually go through exactly what these things bind, okay? So I really do understand what they bind. So our annotations really are pretty real. So how do we use these to get biologically actionable information? So how do we use these to get things that we can actually do stuff with? So this is an example from our cancer cell paper on melanoma. So in this particular paper, we were looking at clinical samples from people, primary melanomas that went to metastasis, and we had both their primary and their metastatic melanoma, okay? And so this is actually the part of the array that was statistically different between primary and metastasis, and it was about 30% of the array, which is actually quite a bit. Actually, I think it may have been even higher than that. But I'm going to show you just one probe in there. So this is LCH and PSA. They bind a sugar called corfucose. That, because corfucose is only made by one enzyme, FuCT8, we already had a target to see whether this was, is this just a biomarker of change, or does this have any real meaning? We were able to look with histology in larger cohorts of samples to show that yes, indeed, we do actually see corfucose go up in metastasis. Ancillarily, we also saw 1,2-fucose go down in metastasis. And so you see the switch that tells you again that the way that these things are connected matters. Fucose is not an all-consuming thing. The way it's connected actually changes its biology, right? And then we did, I'm not going to show you, but there are 2 mouse models in here showing that when you get rid of FuCT8 or lower the levels of FuCT8, you actually inhibit metastasis. So we have 2 different mouse models in this paper. It's a powerhouse paper. And I should say it's with Eva Hernando, who is my amazing collaborator at NYU Langone, who is an amazing melanoma biologist. And so what we did is we wanted to see, like, can we figure out what the mechanism is of corfucose helping metastasis? So what we did is we did glycoproteomics. We put that same probe that we got from the array now onto a column. We pulled down proteins from 3 different commonly used melanoma lines. And we were able to look at what's enriched. And we saw, unsurprisingly, things involved in cell migration. Given that cell migration is a huge part of metastasis, that was not surprising. And I'm going to focus on just one of these, which is L1-CAM, which was an interesting protein that had been found by Massague's lab at Sloan Kettering to be involved in the migration of breast and lung cancer to the brain. Why does that matter? Because melanoma has incredible brain tropism, right? About 70% of people who die of melanoma metastasis have brain lesions, right? So it is highly neurotropic. So it turns out that in that case, what they had shown was that the L1-CAM normally gets cleaved by an enzyme called plasmin, so there's proteolytic cleavage. But in those particular cancers where it went to the brain, antiproteases, an entire class of proteins, prevented that cleavage, right? Melanoma does not have high levels of antiprotease. What it does have is high levels of corfucose. And it turns out that there is a fucosylation site right next to the proteolytic cleavage site. And that corfucose is actually doing the same job as an entire class of proteins and preventing proteolytic cleavage of this particular protein. And that's part of the mechanism by which this is actually helping metastasis, right? We also think this points out the importance of glycoforms, right? Because fucosylated might be a specific glycoform that might be able to tell you more about like what cancers are going to metastasize, right? And metastasize to the brain. So this is just to give you an idea of like and actually this is not a complete list. As I was like, oh yeah, I have more than this. Of what we do, like what we're currently doing. So we're working on multiple sclerosis with Chris Power and Jason Plamel. These may be familiar names. We're working on cardiomyopathy with Gavin Udit. This is a new collaboration that we're just starting. I've been working for almost a decade now with Iman Osman and Eva Hernando, especially Eva Hernando, on melanoma. We have worked in the past on pancreatic cancer with Daphne Barsagi, and I'm really pointing this out because it's one of our recently published papers. Although we're not currently working with Dr. Barsagi, we're currently working on PNENs, which are neuroendocrine cancers, with Sharon Gorski's lab at UBC. We've just started a collaboration with Dr. Dina Kao on fecal transplantation. We have some beautiful data on synovial fluid and osteoarthritis with Heidi Riesink, where we're comparing different models for osteoarthritis to see if we can come up with what will be a proper model to actually look at treatments. So looking at the equine model, a dog model, and humans to see what's common. We've been working on sexually transmitted diseases with both Laura Securo, who we work with on chlamydia, and Eric Arts, who we work with on HIV. We're working on antibody-mediated kidney rejection with Anna Konvalinka at University of Toronto. We have some beautiful data there looking at ABMR and people who do and don't reject in the presence of antibodies to see if we can see differences. And we actually have some. We have a long-standing collaboration on influenza with Elodie Gedden and Stacey Schultz-Cherry. And this is only a bit of it. We've also done work on SARS-CoV-2. We're working on long COVID. Yeah, my lab does a lot of stuff. And I should say the other half of my lab works on microRNA. And so this is actually only the work of half of my lab. It's a little bit nuts, I admit. I looked at it, I was like, oh yeah, we are working on a lot of stuff. So I want to talk to you a little bit though about one of the most common one of the most common things that you look at as clinicians, which is sera, right? And like what sera might and might not be able to tell you about people. So I want to point out that sera is incredibly glycosylated. So yes, 50% of it is human serum albumin, which is not glycosylated, but the other part of it has a lot of sugars. In particular, I'd like to point out that IgG, IgA, and IgM are all glycosylated, okay? And the glycosylation of IgG, as people have been finding with therapeutic antibodies, is incredibly important. That same core fucose molecule that we were talking about in the context of melanoma, if you have core fucosylated IgG, it actually dampens ADCC, which is a problem for immunotherapeutics. So this is something that companies have to be really careful about when they create their IgGs. But you can't make IgG without the glycan. It's actually critical for stability and folding, right? And it's really important for interactions with the Fc receptor. So you have to have the sugars there, and you have to have the correct sugars for this to actually work properly. Also, complement proteins are glycosylated, mucins are in the blood, protease inhibitors are glycosylated. You also have a lot of innate immune lectins, MBL. Again, people who talk about MBL2, they don't necessarily know what it stands for. Mannose, it's a sugar. Mannose binding lectin, that's what the actual name of it is, right? DC sign is a sugar binder, right? MBL2 is a sugar binder. The Siglacs are all sugar binders. Right? So when we're talking about immunotherapies and people are talking about resistance and they're talking about the next generation of check, right? And they talk about the Siglacs, those are all sugar binders. Galectins, sugar binders, right? Many of the things that people are thinking about actually bind to sugars, and understanding their cognate ligands is actually important. In contrast, people who are from East Asia actually have low levels of MS and relatively high levels of this 1,6 branch. So it's possible that we might actually be seeing a marker that might help us to inform us of people who might or might not be more susceptible to getting multiple sclerosis. So that's one example of what serum glycans may be able to tell us. What's an even more clinically relevant example? So what about vaccination? So people get the flu vaccine, right? I got the flu vaccine. Hopefully all of you got the flu vaccine. And, but when you get vaccinated, sometimes there's a mismatch with the flu, and that's different. But even when match with the flu, some people still go on to get flu and other people don't. So how do you tell who's going to have a really good, robust response to a vaccine and who is not? And why is this important? Because let's say that we know that some percentage of the population is not going to respond. Could we give them vaccines that have better adjuvants that make them more responsive? right? We might give them a slightly different vaccine that's tuned to them. Again, getting back to this idea of personalized medicine, right? So that we already do this for people who are older, right? We give them different vaccines than we give a younger population to help stimulate their immune response. But it's possible that that might be true in the general population as well, right? So how do we figure that out? So this is a study that we did with Ted Ross's lab in the civic cohort. So we have 160 adults who received the Fluzone vaccine, the typical shot that all of us get. We have serum that was collected pre-vaccination and post-vaccination, and we have the hemagglutinin assays that are commonly used to assess response. It's a very old-fashioned assay where you literally agglutinate red blood cells, but it's still it's still what's clinically used to look at response. And we have our lectin microarrays. And so we linked the glycosylation profiles to the antibody responses. And this is just showing you that there's a whole bunch of data here. You don't need to pay attention to that. But this is what we actually found. So what we found is in people pre-vaccination, right, so this is before they were vaccinated, people who responded had low levels of these sugars, of these Lewis A antigens. And people who did not respond had high levels of these Lewis A antigens. Why is that important? Well, Lewis A, this set of antigens, is what happens when you have a competition for this epitope, which is type 1 LACNAC, between two enzymes. One is FUCT3, and the other is FUCT2, also known as the secretor gene, right? So people who have low levels of this divert into this pathway, right? And have higher levels of this particular glycosylation. And we think, so there's actually some evidence that non-secretors have lower diversity in their microbiome, right? And there's evidence that higher diversity in the microbiome is actually associated with more effective vaccination and immune response, right? And so we think that it's possible that the low levels of FUCT2 are changing the host microbiota in ways that are changing your vaccine response. And in fact, there's a study in mouse that shows if you feed mice 1,2 fucosylated epitopes, you actually get better vaccine response, right? So if this mechanism is correct, it may mean that non-secretors actually will not respond as well to the flu vaccine, right? And that may tell us that we need to start thinking about stronger adjuvants for that portion of the population. This is something that we're still in the process of trying to study with the NIAID and Elodie Gedden's lab. So another question is, you might ask, okay, so with all of these other omics, how important is the glycome, right? You know, like we have all these omics, like where does the glycome sit in this in terms of telling us something, right? So we have a partial answer to this question by looking at multi-omics of that same sample set. So I was part of a cohort where we actually had a multi-omic team, right? And I showed you our glycomic part, but actually they also did transcriptomics, proteomics, and metabolomics on those same samples. And Sigmund Bayrak in Bin Zhang's lab, who's an informaticist, actually used this information to create 5 different immune types, right, that are related to their ability to respond to the vaccine, right? So high adaptive immunity, low innate, low adaptive high, it doesn't really matter, but there are 5 different types here. And just to show you, this is an example of this. So this is actually part of how they clustered it. And this is actually based on the transcriptomics. So you can see that people who have high adaptive have high levels of the adaptive immune response, transcriptome, and et cetera. So this is sort of how they defined these particular populations. But I'll show you. So if you look at the high adaptive immunity cohort, by metabolomics, you could see that they had markers of inflammation. So arginine biosynthesis is considered a marker of inflammation, platelet degranulation, and synthesis of 5-icosanotetranoic, I can't say that either, acid. I will cop to the fact that I also cannot say that word. So this is what you see in the metabolomics. If you look in the glycomics, we actually see glycan markers of inflammation. And I should say our arrays can also have antibodies on here, so we actually have the D-dimer antibody on here as well. But what we see is P-selectin, which is a lectin that's actually known to be involved in inflammation. Sial Lewis X, one of the most famous carbohydrate markers of inflammation, right, comes up as high in people who have high adaptive immunity, corresponding to what we're seeing in the metabolomics, right? So we actually see markers that, again, are showing are showing us that the glycome is also showing us these markers of inflammation with these people who actually are having high adaptive immunity, are responding to the influenza vaccine. And this is just to show, if you look at the top 10 features for these 5 different sets, right? The one in red is the Glycomics. In 2 of the 5 sets, the glycome actually beat out, so here, like this is almost completely glycomically defined, right? In the top 10 features. This one has at least one glycan marker in it. And what's interesting about this is like we have about 120 probes on our array, some of which are redundant. So we're talking about maybe 60 to 70 real epitopes, right? Metabolome is, they're looking at almost like 1,000 compounds, 40,000 transcripts, hundreds of proteins, and yet our little baby, like our lectin microarray, which is by no means as comprehensive as we would like to make it, is still, because again, integrated information of the cell, right? It's taking all of that information. It's incredibly information rich, right? It's still one of the things that's defining immune response. So just to give you another example of glycans and host response, here's a little bit of our work on SARS-CoV-2. I'll go through this a little bit quickly because I want to leave time for questions. So we saw in the COVID era, we actually were, like many of us, looking at COVID research to try to figure out what was going on with SARS-CoV-2 infection. This is actually from an Alberta cohort. We found that in severe COVID, in the sera, we had high levels of 2,6-sialic acid. Remember that this is not something that in the general population you see a lot of variation in, right? So we got high levels of that. If you look, and this is now from the autopsies done in New York City. These are biosafety level 3 autopsies during the first wave of COVID, right? So these are early on. We saw that again in the lungs of people who died of COVID, they have high levels of that same 2,6-sialic acid, right? And so this is the histochemistry for that, compared to people who died of other respiratory illnesses. When we did the glycoproteomics, we found that complement factors were highly 2,6-sialic acid sialylated in COVID. And actually, it was a glycoform shift. So you could have cases where you have the same level of C5, but the 2,6-sialylated C5 was much higher in the COVID, in people who had COVID. Why is this important? Severe COVID. Why is this important? Because complement was looked at as a biomarker for severe COVID, but one of the problems was not consistent, right? Sometimes it was higher, sometimes it was not. It was not quite consistent. That may be because it's actually the glycoform of complement that's important, not necessarily the complement level, right? So this is why bringing glycosylation in is adding a second level of information to these common protein biomarkers that people look at. And just to show that you actually do see, so this is for C9, you can see that here's the COVID-19, here is the immunohistochemistry of the same patient in both cases, and you can see that the C9 deposition looks a lot like the SNA deposition, right? You're actually seeing that they're overlapping, indicating that this, part of this may indeed be due to complement glycosylation. So, and I'm going to show you, I think I'm probably going to skip my last story because I want to make sure that there's time for questions, but I am going to show you this story, which is an old story that's clinically relevant. This is actually our very first foray into translational medicine, back when our arrays were relatively new. So this was published in Nature Medicine in 2012. And this was actually a collaboration with Rebecca Fitzgerald's lab at the MRC in Cambridge. And so what they were really interested in is, as many of you clinicians may know, people who have Barrett's esophagus, right, which is a condition of the esophagus, have about a 0.2 to 0.5% chance of progressing on to esophageal cancer. And you want to catch that early, because if you don't catch it early, it's a disaster, right? So you want to get it before it goes to cancer. But at that low of a rate of transformation, you can't treat everybody for cancer who has Barrett's esophagus. That makes no sense, right? So what do? Right now, the standard of care is that you do endoscopies and biopsies on a regular basis on people who have Barrett's esophagus. But it's a 0.2 to 0.5% chance, right? That is a horrible amount of money, of time, of resources going in to triage, to monitoring. So can we tell who is and is not going to develop esophageal cancer from people who have Barrett's esophagus, right? And that was the question that Rebecca Fitzgerald's lab really wanted to ask. And she's a clinician, and she wanted to figure out whether she could actually inform clinical practice. And so she came to us and asked us if we could do lectin microarray analysis. And this is actually from, you can see they didn't have very many samples, so we literally didn't have very much to work with, but they gave us, in this case, this is before we knew we could use FFPE, so they gave us fresh frozen tissue. And so they sent it to us, and we were able to do the arrays on it. And we had squamous esophagus, Barrett's esophagus, and low-grade and high-grade dysplasia as defined by histologists. And this is actually the part of the array that looked really different between these. And what we could see is, and I should say this is now low binding is yellow and high binding is purple. So as you go from purple to yellow, you're losing the epitope. What we saw is changes in fucose, where we're losing fucose as these people are going to dysplasia, and changes in glycnac. And so that we're losing glycnac, terminal glycnacs, and as seen by wheat germ agglutinin. And so this is now a little bit of a histochemistry of that. So if you look at the AOL staining for fucose, you can see here's the Barrett's esophagus, a low-grade dysplasia, Barrett's esophagus, low-grade dysplasia, and this is actually what it looks like on a series of clinical samples. Here's the wheat germ agglutinin, you see the same thing. So you can see that as you're going from Barrett's esophagus to esophageal cancer, you're seeing a shift in the levels of both glycnac and fucose. And one of the reasons I wanted to point this out is that I looked in the literature to see, did Rebecca Fitzgerald's lab ever take this on? Because we haven't been involved in this for a long time, right? And what I found is that actually they did. So they went in, so they've been studying this for a while. And most recently, they've been looking in a cohort in the Netherlands, which is a much, much, much larger cohort now, to look at people, these are all Barrett's esophagus samples, but they have the data to know who goes on and who does not. And they have found that AOL is actually predictive. So the odds ratio, if you have 3 or more abnormal compartments, is 4.1 for AOL. It's actually predictive of people who may go on to have esophageal cancer, along with cyclin A, actually P53, AOL, and an expert looking at the dysplasia. Basically, the combination of those 3 actually allowed for predictions of people who will and won't go on. So you could start thinking about, can you use this to now maybe have people who have more constant active monitoring versus people who maybe only need to come in once every few years, right? To be able to see who does and doesn't go on. So this is showing you that you can start to actually figure out who might benefit from more active monitoring, from more active treatments by looking at the glycome. So I'm actually going to skip the entire last section because I think for the sake of time, this is actually our latest paper. I'm skipping it all because I want to leave time for questions. And so I want to just get to my final slides. Yep, it was a bit longer than I thought. Okay, so what I want to say is that really, when we start to think about precision medicine, who will benefit from monitoring? Who will benefit from vaccines? Who will benefit from, a surgery? Who might benefit from an immunotherapy, right? We need information to tell us who will and won't respond well, right? To help inform those clinical decisions. And right now, we've been missing the glycome in this, right? Most people, most clinicians, most biomedical researchers don't even think about it, right? Like they don't even remember it exists, right? But I would argue, based on our own work, that it is actually a really information-dense, information-rich omic that can tell you a lot about the health of people, that can tell you a lot about who might or might not benefit from additional, monitoring, different adjuvants, different medicines. And so we think that this actually, it already is informing clinical practice. Doctors use this constantly in clinical practice already. They just don't recognize it, right? Whether it be blood type or whether it be the, CA19-9, you're already using it in clinical practice. And I think it's really, really important to include this even more as we go forward, because this is an area where we are missing. It could really be beneficial, I think, to medicine. And so with that, I'm going to leave you with the fact that, so this is what we're currently working on. What will we work on in the future? This is part of the point of doing grand rounds is, you might have a question where this might help you, right? And so this might be a great opportunity for collaboration. And so with that, I have to say, and this is the one slide that didn't come through. This is so weird. All of my other slides look exactly like they do on my computer, and the font's all weird. I have an amazing lab. I put the names of a lot of the people who actually did the work on the slides because otherwise I won't remember to thank them all. I have a really amazing set of collaborators as well. This is obviously not the entire list because I would have like slides of it. And this is my lab. They're amazing. I'm really, really lucky to have them. And thank you all for inviting me. I'm really so privileged to be here.
So we're, okay, question, yes.
What a fantastic presentation, and thank you so much. I just, because I have to run, I really needed to ask you, we know that certain things can impact epigenetics, for example, you know, some of the other omics, maybe not genomics, but in general. Right?
I wanted to just stray away from disease states for a second and ask you, what are you aware, or could you share with us, can you impact your glycomics with other measures? So in other words, if you know that there's certain population that's, you know, predisposed because of the glycomics, you know, are there sort of, I guess I'll put out there, lifestyle measures or medication,
There,
other things that can.
yes and no. So it depends on the situation. So the glycome both reports and informs biology. So I think of it as it tells you who you are and how you're doing. And means that all of those other networked things are involved in creating the glycome. So affecting them affects the glycome, right? It's a fundamental part of cell state. And actually I will go to one example, if I can, that actually shows that. No, sorry, I'm going the wrong way. Because I want to just sort of, it's going to get there. Nope, for some reason, this is just not taking me the way I need to go. Oh no, I guess I did have it here. That was actually the one I wanted. Okay, so this is from our COVID study. And I put this up just because I think it's really indicative. These people all died of different things or COVID, right? And what you see is that the heart, kidney, liver, and the upper and lower lobe of the lung all have glycomes that are conserved within the tissue that are separate from, that are actually indicative of the tissue. And I point this out because, one of the things that people talk about is how glycosylation is so variable, but it's not, right? Like this proves that, because you can actually literally tell what tissue it is from the glycome. That said, some parts of it are, right? As you transform, as you change, So in some cases you can. So people are looking, actually, there's a clinical trial right now looking at Glycnac to change the branching state in multiple sclerosis by feeding people Glycnac, right? There, one thought about what you could do if you are a non-secretor, and if the effect really is through the gut microbiome, is to add fucosal lactose, which is actually a common ingredient in baby formula, right, to your diet, because that might change your microbiome. Now, will it? I don't know. Those studies have not been done. I would not be advocating anyone doing that, but there's a possibility that it might, right? So there are definitely direct ways of doing it. But also in an indirect fashion, all of the things that change your fundamental cell are going to change the glycome of that cell, right? So, and in some cases, we think right now there are only very few inhibitors of glycosyltransferases or things that affect glycosyltransferases directly. This is something that my lab is actually also working on with a collaborator to see if we can start to get into this. But I think as we start to realize that these are potential targets, that's another aspect. Right, in cases where core fucosylation is really, really high, would a FUCT8 inhibitor that is targeted to the pancreas, like start to allow you, or not to the pancreas, sorry, to the melanoma, start to allow you to lose metastasis, right? Are there things that we could do? So I think it's definitely a ripe area for the development of therapeutics.
To the mic. Are glycosylations good targets for T cells?
Yeah. So what do you mean by T cells?
T cell receptor.
Do you mean as like CAR T type therapies, or do you mean as like
as a basic physiologic, response?
I can't remember whether the T cell receptors directly. I mean, yes, T cells do recognize glycans in. I think some of them do. You can make CAR Ts to glycans, for example. And people are working on that. I mean, I personally think that the best interactions for that, for things like cancer therapy or other therapies, are going to be the intersection of the glycoform, where where you have it to both the glycan and the protein, and you're taking advantage of the fact that you're double dipping, right? You're looking at two entirely different sets of genetic changes that have to go together, right? So I think that that in the end is going to make things much better. But yes, you can. They do recognize. I mean, antibodies recognize them, so you can make T cells that actually recognize them. I think, but also glycosylation changes. It's funny because we have a paper that's out in with some collaborators that we're on that actually talks about how T cell glycosylation itself changes. And so the T cell glycosylation, for example, is a marker of aging, and that actually has to do with like the way that the immune system works and regulation by Cglex and a lot of other things. So glycosylation plays a lot of different roles in T cell biology.
Another question about, because it seems like these glycosylation signatures are very important, although we may not know exactly what they mean at the moment. But in aging studies and things like that, even in cancer, we know that diet plays a role in therapeutics as well. So that means the types of foods you eat, epigenetics. But I'm thinking more, you know, for people who do, let's say, intermittent fasting or have a specific diet, a ketone-rich diet, so they don't metabolize carbohydrates as much. So does that have an effect on the glycosylation process? So A, the metabolism of, you know, cells that utilize more glucose, but B, sort of diet in general.
So I think we don't really understand that yet. I mean, here's what I will tell you. In cell culture, which we've done the experiments, but we never published, on the overall glycome of a cell, you can change the levels of glucose and the levels of glutamine quite drastically. And until you hit a stress point, you don't really see changes in the overall cell surface glycosylation. Now, if you look at specific proteins, that's different, which I think is part of why people think that glycosylation is so crazy, you know, and so heterogeneous and so like not consistent. And so if you look at specific proteins, you can see changes in specific proteins, but the overall, but like the cell has a concerted reason to have glycohomeostasis. So, and I think the same is probably true in the body. I think as you improve the health of someone, as you change the cell state itself, they change. And I do think that especially in the microbiome, because many microbes, like most microbes use glycosylation and sugars as food, that yes, that probably does change like the cohort of microbes you have, and that in turn will then change your health. And so that's a connection we don't fully understand. Yeah. So there's a lot there, though. We're at the very beginnings of what can be done.
What's the effect of migration? You mentioned about increased risk of MS in European ancestry versus less in European. So did we measure the sample people living in North America, or we measure the sample in East Asian living there?
This is ancestry in this particular case, and we took out confounding factors like so that particular study, which this paper was done, you know, the Snyder Lab really careful in the multi-omics to get rid of confounding factors like people who move versus and there are differences in microbiome and people who moved versus people who didn't move. But the things that we're looking at seem to be independent of, right? So they seem to be more ancestry based. You know, what that looks like in the long term, I don't know. You know, I mean, it's possible that diet and all of that may also have an effect because it does have an effect on autoimmunity. And so how all of that intersects, I think is not really clear.
With that, thank you much, Dr. Mahal. It's collaboration.